Welcome. I am Rick Ludischer. I'm the moderator of the full scientific session, They and We Are What We Eat, linking diet and gut microbiome to human health. Thank you for joining us. This presentation is one of 3 pre-recorded videos focusing on the speaker's material before all of us come together in February for a live panel discussion. In this video, Vincenzo Fogliano will be discussing how food design can impact gut microbiome. Midway through the video, Carolyn Slupski will be joining Vincenzo to further delve into the material.
Thank you, Mr. Chairman. Welcome, everybody. So today I'm going to present to you my lecture titled How Food Design Can Impact Gut Microbiome. The essence of this presentation is summarized in this slide. So basically, we can, my message is that we can design food in order to think to the feeding of the microbiota and not only to the feeding of our body. So basically, the moment that we start to think about the design of a food, we can take 2 different directions. The mainstream up to now is to design a recipe that are full of available lipid, protein, carbohydrate, and phytochemicals. And these are all very easy to absorb for our body. And then means that our body receive usually an excess of calories because it's, but also an excess of nutrients. So in this obesogenic environment, we have human body that become overfed, and the microbiota in the lower gut become starved. But this is only the way we design the food, because we have all the possibility to think to food with where macronutrients has a low bioavailability. So for instance, we can have a low lipid, low protein, and low carbohydrate bioavailability, and also the phytochemicals can be made less bioavailable. So in this case, our body absorbs less of this, and a lot of material reach the microbiota in the gut that thrive on this abundance of nutrients. So this is the overall concept of my story. And now I will explain you basically how we will give you some example of how these translate in practice. So everything is around the digestibility, because of course what is digested is broken in small pieces, become bioaccessible, and then generally bioavailable, so it's taken by the body. What is not digested become less bioaccessible, not bioavailable, and is reached the lower gut. So I will give you now 4 examples of lipid delivery, oral processing, the role of plant cell wall, and the story of polyphenols. About the lipid digestion, I want to give you this example by my co-worker Eduardo Capuano, and he made this study with hazelnut, and working with roasted and raw hazelnut, and playing with the particle size. So you see that when the particle size are very fine, you have the maximum digestion of the lipid. When they become more coarse particle size, and especially if we do not have any roasting, the lipid are digested at much lower extent. So this means that all these lipids reach the microbiota, and bacteria can grow on these nutrients. Another examples I want to give regarding what happens in the mouth, the oral processing. So this is something that we can easily modulate with design. And I want to show you this example made by one of my PhD, Arianna Van Eck. She works with carrots, and she cut those carrots in cubes or julienne. So when you provide people these carrots, they take much more time to masticate the julienne carrots. That is not obvious, because actually the cubes are much less pieces, so looks like less processed. But it's actually the case. So, and when you look at the bolus, so the bolus after the mastication, you see that in the julienne carrots there are many more particles. So we can expect that there is more digestion and more material would reach the gut. You can play many examples around food processing, oral processing, sorry. And this will always be the case that you can play with the time of processing food. So with what we call the eating rate, that is a very important parameter to consider when we want to influence a parameter like satiety, the sensory, and also the delivery of nutrients to the gut. So the oral processing is one of other weapons that we have in our possibilities. Third example I want to give you regard the plant cell wall. We can use plant cell wall, intact plant cell wall, to limit the digestibility of the macronutrients that are inside the cells. The most classical example regard the starch. So basically, if we have granules of starch within the cell walls, these granules are not easy to be reached by amylase, and then glucose is not released and stay inside the cells. So when then these plant cells reach the gut microbiota, then there the bacteria has plant cell wall degrading enzymes, and they are able to access this starch that is within the cell, and they can grow on it. And just to give you an example from one other of my PhD, Anna Rovalino, you can see how with beans, she was able to obtain beans, cooked beans with intact cells, with all the granules of starch within the cells. And when you look at the hydrolysis of starch from these different damaged red kidney beans cotyledon cells, you can see how the starch can be completely released or just released much more with a much slower kinetics. So this is also an interesting approach. And now we are repeating the same type of approach with proteins, because also proteins, traditionally, we want to have the maximum digestibility of proteins. So all the amino acids go to the body, but actually proteins are also useful for our gut host. So in this experiment, Jan Zhang, how he's working with different source of tryptophan, because we are interested in look at what is the fate of tryptophan in the gut. So how the gut microbiota use tryptophan, because we know that there is an activation of these aryl hydrocarbon receptors that is an important mediator of inflammation. So Jan provided the tryptophan as a free amino acids as a part of a soybean protein isolate, or of intact cells again, or even clustered cells of soybean. So different type of accessibility of the tryptophan. And he will look at all the metabolites that are generated by the gut microbiota in the gut. And you see that now, I have no time to explain, but the kinetics of release of the different gut metabolites are actually in line with the source. So when it's a free tryptophan is very fast. When it's a clustered cells with tryptophan with the protein inside, it's much slower. And this is not only an importance for the kinetic of release of these metabolites, but also for the activation of AHR. And so the biological effect of the protein digestion in the gut is very important for the immunomodulation. Last experiment I want to show you, last case, regard the polyphenols, the phytochemicals. This is a kind of historical work for my group. So polyphenol in the food can be in very different forms, so free or chemically bound or entrapped in the matrix. So and it's clear that although in general phytochemicals are not very bioavailable, but when they are entrapped in the physical structure of food or covalently bound is even less. So we published already now 12 years ago a very important paper on trends in food science about the importance of the fibers to deliver polyphenol to the gut. So the fiber was not only important for its role as a fuel of the gut microbiota, but also because deliver the polyphenol to the gut, and the gut actually release polyphenol in the blood. And in this case, we do not only have in vitro evidence, like in the case above, but we did human studies. An old one, where that kind of serendipity discovery, we found in this study with a group of Glenn Gibson in Reading that the feeding people with whole grain or wheat bran, we were able to see an increase of ferulic acid, so the phytochemicals in the fasting subjects of fasting, in the plasma of the fasting subjects. So there was the idea of the cartoon I showed in the previous slide, that the fiber bring polyphenol to the gut, and the microbiota release the polyphenol. And many years later, so in 2015, we were able to publish that actually an intervention study with the whole grain wheat consumption is able to increase the ferulic acid metabolites in the blood. So we were able to demonstrate exactly the concept and the importance of the phytochemicals. So to conclude, the take-home message of this presentation is that the food design can be used to modulate the amount and the kinetic of the food component release in the various gastrointestinal compartment, and especially on the gut, and so feeding the gut microbiota. And in this way, we can improve the health of the consumer by designing a healthy food that considers the complexity of the gastrointestinal environment and the digestion machinery. And in this way, we can start to bridge the gap with the famous story of the personalized nutrition. So we can provide food that are designed for the personalized need of the consumers. Of course, this is just the beginning, but I think is an exciting field for everybody who's interested in healthy food and the effect on public health. And with this, I would like to thank all of you for the attention.
Thank you, Vincenzo. Now, Carolyn Slupski will join the conversation. Welcome, Carolyn.
Fascinating. Thank you for that most enlightening talk. I really like the idea of really looking at the food matrix and actually working with the food matrix, I think, to optimize delivery of nutrients. So have you looked at ways that you could actually do this? Like, I think a lot of what you talked about was specifically on the whole foods. Have you looked at ways that you can do this with just processing?
Let's say that our preference now, the line of research we are doing is indeed try to work with the whole food, to also to take into account the preferences of the consumers, the concept like naturality and this type of thing. So it's now, but you are right that there are many other tools and processing tools that we can use to make ingredients that actually are tailored for the delivery of compounds. So in the past, we have done very nice results we obtain using ingredient encapsulation. That is, of course, a very powerful tool that you can adopt to schedule the delivery of the compound in the gastric environment, in the duodenum, in the ileum, or in the gut. So it's really, you can, and that's a fascinating possibilities. But let's say for the proof of concept, I prefer to use this for proof of concept, because at the end, encapsulated ingredients turns often to be very expensive and not realistic to use in the real food. So if we are able to play with the food matrix as it is, I think is most preferred choice at the moment.
Okay, great. Yeah, I was particularly interested, I think, in when you were talking about specifically the tryptophan. And I think you were showing that, um, and actually, maybe you could describe that a little bit again for me, because I wasn't quite clear. Was this tryptophan through a specific protein that you were providing, or um, maybe you could give me a little bit more information on that?
Yeah, I apologize. I went fast because of the 10 minutes. Yeah, we selected soybean proteins that are a good source of tryptophan. But we do believe that the approach is more or less comparable when you have tryptophan from other sources. So the point is that yeah, the digestibility of the protein at the end influences the release of tryptophan in the environment. And so that's the first step. But the second step is also the accessibility of the proteases to the protein is going to change the kinetics. So if we keep the proteins confined into the cell walls, we know that proteases take time to reach it, and then the digestion start at a much slower pace. And this is especially now with the booming of the plant food and the replacement of animal protein with the plant protein. I think this is something that we have to take into account. The kinetic of the release of amino acids for the gut microbiota.
Okay, and so I guess with the soybean, and just so I kind of understand that a little bit. So so the protein is actually bound up inside the cell of the, I guess you provide whole soybeans, or is it a soybean processed product?
Yeah, we isolated the cells of the soybean. So basically, it's very simple. It's not a rocket science. You just boil the whole bean, and then you mash it. So mashing after boiling, it's in legumes especially, you are able to obtain isolated cells with the intact cell walls. But if you just grind the beans dry, then you destroy everything, and you have a fine flour with all the cell walls are broken. So our direction is to work in the isolated cell from legumes. Isolated cotyledon cells is called. Yeah.
Right. Yeah, very interesting. Yeah, I mean, yeah, just really, I mean, how you treat the cells and the food, and I think it was kind of also indicative. What I was really quite amazed at was the work you did with the carrots, that the julienne carrots actually produced larger particles than the cubed carrots, and in fact, people were eating them or chewing them. Were those were raw carrots, or were they cooked?
No, that one was raw carrots. It's like a coleslaw salad. That's was what we want to mimic. And indeed, at the end, it's kind of paradox, because if you make a complete puree of the carrots, that become very fast, because it's you swallow immediately. But at a certain point with this intermediate processing, because julienne is you can make very small julienne, but people is forced to take it longer in the mouth, because it's the time that your tongue is able to make a coherent bolus, and that's actually a lot to learn in how the swallowing time is gonna influence then the eating rate, and then the digestion. It's all connected, and it's the fact that for me is fascinating. That is something that we can really do as a food scientist. So it's not so if we treat the mastication and the digestion as a fixed machine, because it's more or less similar for everybody. There are difference, infant, adult, and so on, but it's a fixed system. But the way we put the food in the mouth of the people, that's something that we decide as a food scientist. So it's the moment where a food scientist can really influence the actual health outcome of of a certain meal.
Right. Yeah, no, absolutely. I think that this is absolutely fascinating. And the work also you did on the whole grains and looking at the polyphenol release as well. I think that that's really important. I think people have been trying to develop things like supplements and really just trying to provide these pure compounds. And I love this work that you're doing, because what it's showing is yeah, I mean, when you just have those pure compounds, or you don't prepare the food in the correct way, that yeah, it's not going to reach those gut microbes and influence them. And we know that the polyphenols have an influence. We know obviously tryptophan is huge because it makes molecules that are really important for gut health and so forth. So I think, yeah, this is all absolutely fascinating.
Yeah, that was actually what I love of that study, that we started with a screening of product on the market. So, and just looking at the product on the market and the cereals product, some of them, they have plenty of free polyphenols, and others are more with the bound polyphenols, so bound to these matrix. And at the end, I mean, those that are, let's say, labeled as more healthy are those that where there are free polyphenols. There are many studies also to increase the amount of free polyphenols. And we wonder why we have to create this bomb of free polyphenol that at the end is also you have when you have coffee or tea. I mean, there is plenty of free polyphenols. Mm-hmm. So in cereals, it's the other way around. In the whole grain, these polyphenols are bound to the matrix. So what happened if we investigated that? And because it's true, they are not bioavailable, but they are useful for the gut bacteria. So this was the let's say the idea at the beginning, and indeed at the end we were able to prove this concept that I think it has long roots in the research. During the 90s already, some scientists investigated this concept, but I think that now the time is because this story of the microbiota and feeding the gut is something that now is mature. So we can look at this as usual happen in science, looking at the results in the past and reinterpret it a bit, considering the view that we have now on the food and health matter.
So in the future, do you think that you're going to go into looking at mixed feeding? So for example, looking at your coleslaw salad with different types of protein, and look to see how absorption and so forth are optimized in a mixed meal?
Mm-hmm. Yeah, this is really fascinating. I agree with you, Caroline, that's really the future is to study this mixed, the composite food, how all these interaction results at the end in the real, dietary, in the real diet of the people. That's complicated because we want to reduce to something that we can understand, and the more we make things complicated, the less we are able to understand the cause and the effect, but it is the direction to go. So now we have so many tools to investigate how high throughput and this type of thing. So I think we have to go in that direction, but let's say there is work for the next 30 years when you start to mix and to create this composite story.
Yeah, well, I think that would be an absolutely fascinating way to go, only because, and I sort of have been thinking, I don't know if I want to say anthropologically, but as you sort of think through the ages and how humans have developed in their eating, and it's really in the past, I don't know, 60, 70, 80 years that we've really changed our eating habits. And, a lot of it has been through the development and food processing and things like that. But really trying to understand and look at, because our ancestors knew how to put food together to ensure that we remained healthy. And I think for some reason we've sort of forgotten some of that. And I think that's kind of where we are right now. We need to sort of understand a little bit better, I think, the food and why it is that we always put certain foods together when we eat them. Anyways, I love your work. Thank you so much.
Thank you. And I also like this story of the anthropological and how the mankind evolved. I think that would be very nice to have a session of IFT maybe on this aspect, because there is many interesting things to talk about. And thank you for
Yes.
your question.
Thank you again, Vincenzo, for your remarks, and Carolyn for joining in the discussion. Just a reminder that this is one of 3 spotlight videos focusing on the individual work of the speakers before we all come together in February for the live panel, They and We Are What We Eat, linking diet and gut microbiome to human health. Please check the AAAS annual meeting website for the panel's date and time. Again, thank you, Vincenzo Fogliano, Carolyn Slupski, and all of you, the audience, for joining us. We'll see you live in February.